We'll have a look at agglutination test, specifically Rose Bengal plate agglutination test, also known as RBPT. RBPT is a test that is used to carry out to screen antibodies against Brucella abortus or Brucella melitensis, and this test makes use of colored antigen, and that antigen is called as RBPT antigen, and the dye that is used to color the antigen is Rose Bengal. That's why it is called Rose Bengal plate test antigen. It is an inactivated antigen which has been inactivated using phenol as a chemical for inactivation. It is particulate or insoluble antigen, hence it is used in agglutination, not in precipitation. It is a screening test, initial screening test carried out on individual serum samples, unlike milk herd test, which is milk ring test, which is a herd test. It is very quick and highly sensitive and economical test to carry out. But there are some issues with the test, that it can give you false positive, especially with gram-negative bacteria. So the antibodies against E. coli, Yersinia, Francisella, Salmonella, etc. may react in this test and give false positive. Hence, it's need to be confirmed with more specific tests like CFT and ELISA. This test can be done on clean glass slide or on plates or in tubes, but today we'll use slides. So you need antigen, which is in this vial. You need positive control and negative control, and your test sample, that is test serum samples. To carry out this test, you also require microtips and micropipette, 100 to 100 microliter or 20 to 200 microliter micropipette. And these are wooden applicator or toothpicks that are required for mixing. So this is RBPT antigen. This is pink color, inactivated Brucella buffered antigen. So take a clean slide and mark with positive control, negative control, and with your test sample. Now apply 30 microliter of RBPT antigen onto the predefined spots, which are like for one for positive control, another one is for non-negative control, and third one is for your test serum sample. We'll add now one by one equal amount of different serum samples. So first goes non-positive antisera. This antisera has got antibodies against Brucella species, and then we are adding 30 microliter of known negative control serum sample. It does not have any antibodies against Brucella species, and this one is the control, this one is the test sample. So this is the sample we are testing today, whether it contains antibodies against Brucella species or not. With the help of a toothpick, mix them gently, antigen and serum. Do this for all the samples. Make sure to use different wooden applicator, or you can use even a tip to mix them, or a loop to mix them. Lift the slide, gently rock it and rotate it to ensure that antibodies and antigen interact very well. And you can see in the positive control, you already see some clumping happening, but we will wait for 4 minutes until we read this test, because depending upon the concentration of the antibodies, it may take time anywhere from few seconds to few minutes. So we'll wait for 4 minutes before we declare the sample to be positive or negative. But definitely we see that positive control is showing agglutination. So after 4 minutes you can see that positive control is showing large amount of clumps, aggregates, whereas negative and the test sample are not showing any kind of agglutination. So test sample is as good as negative control. So this means the sample that we tested today is not positive and it is negative. So very important, you see again positive control, negative and test sample. Thank you very much.